Welcome back, everybody. Today we're going to be discussing the most basic form of genetics, which is Mendelian genetics, named after Gregor Mendel. And this video is extremely important because it sets up a lot of the terminology and the basic background information that's required to understand the entire rest of the unit. So without this basic understanding, you will be completely lost for the next month of class. Any conversation about genetics has to begin with the most famous person that really discovered most of these patterns of inheritance, who was Gregor Mendel. And as you probably have heard of, he was an Austrian monk, and you really don't have anything better to do but, I guess, grow pea plants all the time and monitor how traits are passed along from the parent pea plant down to the offspring pea plant. And he's most famous for developing his 2 laws of inheritance that we'll go through at the end. And he was the first one really to describe how traits were passed along from the parent to the actual offspring. And even though he did his work in the mid-1800s, he was not really recognized until the turn of the 20th century. So it was a very long period of time where he collected and journaled all of his entries, but no one really recognized what he was doing or that he was actually correct. So between 1856 and 1863, Mendel, you can see, cultivated and tested some 28,000 pea plants. Because once again, what else are you going to do as a monk? And he found out that the plant's offspring retained the traits of the parents, which seems like pretty common sense that you get your traits from your parents, but really it wasn't at this moment in time. And because of these studies and because of what he discovered in terms of how traits are passed along, he has been dubbed the father of genetics. Mendel stated that the physical traits are inherited as particles. Now remember, at this point, DNA, cells basically were just really being found and being able to be visualized. So DNA was still 100 years away from discovering it. So he had this idea that there's something that's being passed along from parent to offspring, and he called them particles. Now, as we know that these things that are being passed along are the actual genes that are stored directly on your chromosomes that are being passed along through creation of the sex cells through meiosis, and then eventually the combination during fertilization to create you. Remember that genes are these segments of DNA that code for a particular protein. So that's why it's genetics, the study of genes. Some terminology for this unit: a trait is any characteristic that can be passed from parent to offspring. So it could be hair color, eye color are the 2 most common ones we deal with. It could be a genetic disorder. So it's anything that physical characteristic that can be passed. Heredity is the actual passing of these traits from parent to offspring. And then genetics is the study of heredity. So genetics is the study of how you actually how the trait move from the parent to the actual offspring. There's 2 different types of genetic crosses that we'll deal with. You have a monohybrid cross, which involves a single trait only, which is the most basic form of just looking at if one flower is red and one flower is white, what's the chances of you having a red flower as an offspring? So it's a single trait, so it's a monohybrid. Mono means 1. And more complicated is looking at 2 traits, so a dihybrid cross, where you're looking at, well, what's the chances of an offspring flower being white and it being a tall plant? So you're looking at 2 different traits. And both of these crosses, all crosses, are really solved with a Punnett square, which you have definitely used before. As we said, Punnett squares are critical to solving any of our genetics problems. Alleles. So alleles are 2 forms of a gene. So a gene is a gene. So let's say you have a gene for hair color. Alleles are 2 different versions of that gene, kind of like Pokemon games. If you play Pokemon games, you know that there's the base game is pretty much the same, but there's 2 different slight variations of that game, such as Pokemon Red and Pokemon Blue back when I was a kid. Same basic game, but slightly differences. Well, that's what alleles are. They're 2 forms of a gene. So you have one gene, let's say, that controls your hair color. You have a brown version and you have a blonde version. Those are what we call alleles, slightly different versions of that same gene. One of them usually is dominant, which is what we say is the quote-unquote stronger of the 2 genes, and is always going to be expressed. That's why it's dominant. And we represent that by a capital letter. The other form of the gene is usually recessive, so it shows up less common because it is recessive, and it's going to be masked and hidden when there's a dominant gene is present. So if you have a dominant gene, usually it's going to be expressed. And if you have one copy of the dominant and one copy of the recessive, the dominant is going to mask that recessive. And the recessive we represent by a lowercase letter. We have genotype versus phenotype. The genotype is literally your combination of genes. So it's the combination of DNA, the combination of chromosomes that you actually have. So you can see examples. It could be two dominant genes, capital R, capital R. It could be a dominant and recessive, capital R, lowercase r, or it could be two recessive copies of that gene, little r, little r. Your genotype determines your phenotype, which is your physical characteristics. So your genotype will always be a combination of alleles, while your phenotype will always be the physical characteristic. Do you have a disease or do you not? Are you red or are you white? Are you blonde or are you brown haired? So genotype is the actual combination of genes, while your phenotype is the actual physical feature that we see. Phenotype, physical. Genotype, genes. So if we look at an example of this, we have two different alleles. We have capital R represents red coloration in this flower, and lowercase r represents the yellow flower. So if we look at the possible genotypes, we could have two dominant copies, big R, and because big R is dominant, if you're going to have a big R, it's going to show up. So you can see the phenotype of that flower will be red. If you have one big R, one little r, you still have a dominant copy of that gene, so it's going to show up, and you still have a red flower. And if you have two lowercase r's, little r, you have two copies of the yellow gene, therefore your flower is going to actually end up yellow. And these genotypes can be homozygous or heterozygous. So homo means the same. So homozygous means you have two copies of the dominant gene or two copies of the recessive gene. So you can be homozygous dominant, capital R, or homozygous recessive, lowercase r. Homozygous is also known as purebred. So they are interchangeable, and of course I will interchange them to try to confuse you. So homozygous means purebred, means you have two copies of that allele. Hetero means different. So heterozygous means you are a hybrid, which means you have one copy of the dominant and one copy of the recessive. So it can be homozygous dominant, homozygous recessive, or heterozygous for your genotypes. And when we're talking about crosses, we have to label the different generations when we're talking about mating and forcing organisms to have intercourse, basically. So the first generation is known as the P1 generation because that's the parental generation. When you breed the P1 generation, you end up with the first generation, which we call the F1. So the F1 is the kids, the offspring of the P1 generation. And then if we breed two F1 generation plants together, we end up with the F2 generation, which is the second breeding generation. So it's always P1, F1, F2, F3, and so on. So P parental, and then we count F1, F2, F3, depending on how many different crosses, how many different levels and generations we're actually breeding to determine what we're trying to solve, to determine what the answer is to whatever question we're trying to solve. So we look at that if we follow the generations. We have the P1 generation, which is a homozygous dominant tall plant being mated with a homozygous recessive short plant. When we mate them, we end up in the F1 generation, the first generation of offspring, all being heterozygous, so they actually end up all being tall. If we mate two of those F1 plants together, so yes, we're basically mating siblings, brother and sister together, we are taking two hybrids and we're mating them. In the F2 generation, you can see we end up with 3 tall and 1 short due to the end result. So we have 1 homozygous dominant, 2 heterozygous plants, and 1 homozygous recessive. The final thing in this video that I want to discuss is Mendel's laws. So Mendel came up with 2 different laws. His first law is the law of segregation, which says that during the formation of gametes, the 2 alleles are actually separate from each other. And we already know this. We did meiosis already. So you know that during meiosis 1, that specifically during anaphase 1, those homologous pair of chromosomes separate. Once again, Mendel didn't know what chromosomes were, so he didn't know that they're actually physically were separating. So if we look at the example here, we have this dog with 2 recessive genes. Well, during meiosis, 1 chromosome will go to one sex cell, 1 chromosome will go to the other sex cell, the law of segregation. So you start out with a diploid cell, and you end up with 2 haploid cells, each with 1 copy of that gene. You start out with 2 copies of the gene, each sex cell ends up with 1. And then these sex cells are combined with another organism's sex cells, and then you end up with the actual baby. So it's recombining them. Another example of this, if we have a male red flower that is heterozygous, during the law of segregation says that during meiosis, one copy of that gene will end up in one cell because that is going to be one chromosome, one copy will end up in the other cell, and we have a female white flower, so one copy will end up in the egg cell 1, and one copy will end up in egg cell 2. And then if we actually combine them, sperm 1 had a capital R, it's a haploid cell. Sperm 2 had a lowercase r, and all of the female egg cells only had lowercase r's because the female only had lowercase r's to give to their sex cells. And then if we combine them, we can see we end up with 2 heterozygous offspring and 2 homozygous recessive. And if we look at the phenotype, capital R represent red, lowercase r was white, red is dominant to white. So we have one uppercase r, so this guy would be red. This flower would be red also, and 2 lowercase r's, these would be white. And then the second law that Mendel came up with was the law of independent assortment, which said that alleles for different traits are distributed as sex cells independently of one another. In other words, your hair color doesn't affect your eye color. And we also kind of already know this because if you talk, when we did random assortment during meiosis, when those pairs of chromosomes lined up in that double file line, just because the orientation of homologous chromosome pair 1 didn't affect the orientation of homologous chromosome pair number 22. So it didn't matter which way they organize each other. So when they split, just because mommy's chromosome number 1 went to this sex cell and daddy's chromosome 22 went to this sex cell, chromosome 1 had no impact on what was happening with chromosome 2. And that's what the law of independent assortment says, that each chromosome doesn't impact each other. They don't come together. It's not like you get all of mom, all of dad. It doesn't matter. It's random which one goes where. So law of independent assortment really is shown in a dihybrid cross, where you're talking about hair color and eye color. One does not affect the other one. Now, I will say the law of independent assortment is not 100% true, because if 2 traits are on the same chromosome, well, you physically can't rip apart those traits. So if you have your eye color and your height both on the same chromosome, well, they can't be independently assorted because they're on the same chromosome, and we actually call that linkage. And that is it for this video. So that was the entire point of this video, was just giving you some background information, some terminology that you're going to be hearing a lot over the next few weeks.